Hello everyone, and sorry I am so late today. I had trouble getting into Collaborate. Collaborate wouldn't work properly for me, so I apologize for that. Housekeeping stuff real quick. You have your lab practicals happening on April 30th, and that's really all I really want to say to remind you of. So lab practicals happening April 30th. For your homework, you were supposed to watch a video that I sent you. And from that video, you should be very familiar with the term called a cloaca. So what is a cloaca? Yeah, a cloaca is basically what we all had in embryonic development until we were about 3 months old before we differentiated into male or female. And that was just the exit way for sperm, ovaries, eggs, babies, whatever you want to call it, what they're supposed to come out of there, urine and feces. So there are lots of things that were coming out of this one tube. And we also looked at how that area was differentiate, either form the structures for a female, the external sexual structures for a female, or the structures for a male. So I thought that was a really good video, and you should keep in mind that term cloaca, and you should be familiar with the stuff that happened in that video because that's fair game for anything that could be on your test about it. Capacitation is the process of making the sperm do their thing. On Monday we talked about spermatogenesis, and we talked about how that process involves both mitosis and meiosis, and that the first step going from the stem cell for spermatogoa to a primary spermatocyte was considered a mitotic event, and going from a primary spermatocyte to a spermagonian is a meiotic event, which includes 2 divisions, and the process of spermogenesis gave us the actual sperm, and that there had to be some changes that were taking place, and the involvement of nurse cells, and even now we still haven't made these sperm or spermatozoa completely motile and doing their thing. So in capacitation, we're giving the sperm the capacity to do what they need to do. The first step of capacitation is allowing them to become motile, which means to give them more movement. So they're going to mix with some of those sugary secretions from the seminal glands to power their flagella to have the energy that it needs to make its way to the vagina. You also have to make the sperm capable of fertilization. So when they're exposed to the female reproductive tract, they're surrounded by their seminal fluid that helps to not only give you the energy that they need to make this arduous journey up to the upper 1/3 of the fallopian tube where that egg may or may not be waiting. And it also provides them with a buffer or a shield that they're more alkaline or basic in nature to prevent them from the acidic environment of the vagina. So those accessory glands that make the semen, they're formed from your seminal vesicles or seminal glands, prostate glands, and your bulbourethric glands, and they each have a very distinctive biochemical characteristics that are meant to aid and assist the sperm in making their way to fertilize the egg. Major functions of these reproductive glands is that they help to activate the sperm. They also provide nutrients, as we talked about before, that are needed for motility, namely in the form of sugar or glucose. They help to propel the sperm through those fluids by making the reproductive tract, it kind of makes it more slippery, if you will. So it gives it a nice fluid medium. It reduces frictions for the sperm to make their way. The peristaltic contractions during ejaculation help to propel the sperm from where they are out through the urethra. And then, as we've talked about also, that they produce buffers that will counteract the acidity of the vagina and even of the urethra that they're going through, because remember, urine passes through here as well. So we really want to kind of buffer or protect these sperm from the acidic environment of both the urethra as they're traveling out of the male and those found in the vagina as they're making their way to fertilize an egg if one's hanging out up there. Male external genitalia, penis, tubular organ, which the distal portion of the urethra passes through, conducts the urine to the exterior of the body, and it also introduces semen into the female's vagina. And just as the video showed you on Monday, that males have, I guess, 2/3 of a cloaca. They have a separate hole for their feces, but their urine and their semen are going to pass through the same hole. For females, for women, we don't have a cloaca-esque anything anymore. We have one hole through which babies come out of, or our modified version of our egg now that it's been fertilized. We have a separate hole for our urine to come out of, and then we have a different hole for our feces to come out of. So males kind of have like 2/3 of a cloaca. Testosterone is the hormone that helps to nourish, or helps the sperm to be made. It's kind of like a trigger hormone that helps the sperm to start that process of spermatogenesis to take place. It also establishes secondary sex characteristics, so the distribution of facial hair. Once testosterone kind of kicks in puberty, around the time of puberty, you start to see these young males develop facial hair. They also increase in muscle mass and body size. You'll notice that in like the 7th grade, the girls are sometimes taller than the boys are. They're all about the same size. And then all of a sudden in like 8th grade and 9th grade, there is a very huge difference between the girls and the boys, because the boys are developing their body mass. Sometimes boys will even have stretch marks on their shoulders and on their buttocks because they're growing so quickly, so their skin doesn't keep up as quickly with the dividing, and it stretches the skin out a little bit. The deposition of body fat into various places on their body, that also is going to change as a result of testosterone. And it maintains those accessory glands and organs of your reproductive tract, which we talked about what the seminal vesicles do, so making that seminal fluid from the prostate gland, the bulbourethra glands, and your seminal vesicles, and all of those things that we associate with the male reproductive system. It's the job of testosterone to maintain those structures hormonally. Descent of the testes, this is part also within the video that I had you guys watch. Remember, it says the gonads, testes are eggs for males and females, so little baby girls and little baby boys would, by the 3 months embryonic development, before that time, they're just gonads, and they're going to be in the abdominal cavity, and they're right next to the kidneys. There are different hormones that will cause the testes to descend. Testosterone is one of those hormones, and then we had the Mullerian inhibiting factor. It's another factor or protein or hormone that's responsible for making this testes descend outside of the body. The connective tissue extends outside of the peritoneum, so you form these little pockets through which the testes go into. The SRY gene, or the sex-determining gene, also makes sure that this process takes place and descends the testes outside of the body. For people that have a chromosomal abnormality, which means they may have one X chromosome, or their Y chromosome isn't quite right, or they're missing a gene off of their Y chromosome, that SRY gene, then that can prevent the testes from descending down into where they need to be as well. So during the 7th month, the fetus grows pretty rapidly. The circulating hormones stimulate the movement of the descent of the testes. Each testes has to move through abdominal musculature, and it's accompanied by this pocket that's been created in order for the testes to reside in there. So here we have the descent of the testes taking place, and as it goes down into the scrotum. And then here are those gonads. Remember we talked about at 2 months, prior to 3 months, we all look the same, male and female. Then you start to see them form the epididymis on top of them. This mesonephric duct is going to change and become a vas deferens. And even at 4 months, they're still not completely descended out of the abdomen, but they're definitely testes. And with the help of hormonal control, they'll continue to descend into the scrotal sac. And here's just a bigger picture of that. Hernias are protrusions, and these are notes that you don't have here. They're just like extra things that went along with the what we looked at before that I'm going to address real quick here, and then we'll move on to get back on track with the notes that you have. Male hernias are protrusions of the visceral tissue into the inguinal canal. So in your inguinal area, you can that groin area. Spermatic cord can be enclosed in there and can cause weak points in the abdominal wall. A lot of times for babies that are born, males that have a hernia, they leave it alone until they're about 5 or 6 years old, because usually just the strength that's generated by those abdominal muscles is enough to close that up. Female hernias are usually much smaller. The ilial inguinal nerves and round ligaments of the uterus may be protruding out of that hernia. My little baby Eliana, she has a little bit of a hernia, but the doctors aren't really concerned with it. And in fact, even as she's 6 months old and she's learned to sit up and she's been more motile and active, her developing stomach muscles, or she's getting more abdominal musculature that's helping to close that area up. But if she's like 5 or 6 years old, or maybe even up to 7 or 8, depending on the physician and the parents, we may decide to have that that hernia surgically addressed. But chances are it'll probably, as her stomach muscles get stronger, they'll probably close up on its own. All right. So this is a really cool picture that's showing that process of spermatogenesis. It's looking at mitosis and meiosis, actually. That in the stem cell of the sperm, we had to go through mitosis, and you had 2 daughter cells that are going to be exactly the same. Now, when we go through meiosis, and meiotic division, we're going to have synapses take place over these tetrads. So these pairs of chromosomes that look kind of like one another are going to swap some genetic information. And in meiosis, we have not just one area of crossing over that takes place in one division, but we also have another. So that you end up with these gametes down here that are genetically different from one another. So for spermatogenesis, remember there's spermatogonium, and that became to state as another spermatogonium, and then a primary spermatocyte. And then we have meiosis 1, so that primary spermatocyte goes through meiosis. In that meiosis 1, very important thing that happens here is that we have crossing over that takes place, so that these daughter cells are going to be slightly different. So each one of your secondary spermatocytes, they won't have the same genetic composition, but they're going to only have 23 pairs of chromosomes, because what meiosis 1 does is that it reduces the chromosome number in half. So if we go back here, the spermatogonium has 46 chromosomes. This guy has 46, that guy has 46. This guy here, at the end of meiosis 1, is going to have daughter cells that only have 23 chromosomes. And then in meiosis 2, the secondary spermatocyte enters that, finishes meiosis because it has 2 divisions. Then you're going to have 4 spermatids, which are also going to be haploid. They also have 23 chromosomes. And then these 4 spermatids go through the rest of that process of spermogenesis, which is basically just making the sperm do what they need to do in order for them to be ready to go and do their job and fertilize an egg. So spermogenesis is, as we said before, just a physical maturation of the sperm itself. Last step of spermatogenesis, and spermatid gives a single spermatozoa or a sperm. And it takes about 5 weeks for this to take place, but the important thing to make note of is that there are 23 chromosomes in each one of these sperm. And those 23 chromosomes are going to be different from one another. So in theory, no 2 sperm should be alike. Now for oogenesis, this is a little bit different, and I wanted to show you these images before, which are not in your notes, but they're in your textbook, because I wanted to make sure that you understand that spermatogenesis is going to give you 4 functional sperm. They all have 23 chromosomes. They all can do their thing. Oogenesis, on the other hand, is a little bit different. You're not going to get 4 functional eggs from oogenesis. You only get 1 functional egg. So like spermatogenesis, oogenesis starts with an oogonium, and that oogonium is diploid, which means it has 46 chromosomes, which is true for spermatogenesis and for oogenesis, that they have the stem cell has 46 chromosomes, but different from spermatogonium. The oogonium, or their female reproductive cell, they complete their mitotic divisions before birth. So we have our oogonium, and then we have an oogonium that's just going to lay there. So this is just going to, it eventually will go through primary oocyte, oogenesis, I should say. But this guy here, you finish making them. So this guy that goes through mitosis, I'm going to go back a little bit. Not that far back. Remember we said that this spermatogonium it divides, one stays a spermatogonium, the other one becomes a primary spermatocyte. Well, in oogenesis, this process of the oogonium and the dividing, that part takes place before birth. So in essence, ladies, we have the number of eggs we're going to get for a lifetime. So you're not going to see this guy continue to like divide and M, make one of you, divide and M, make one of you, divide. And so again, this guy will divide and make two more, and then that one of those will divide and make two more. That doesn't happen. So that's why there is a reproductive time for women. For males, now as they get older, their sperm aren't going to do what they're supposed to do all the time. They may have two tails, they may swim in circles, they may be really slow, they may have some other things that go on with them. But in theory, males can continue to produce sperm throughout their entire lifetime. So you can be 90 years old and have a 27-year-old bride that you impregnate at 90 years old. But for women, highly unlikely, unless you still got some eggs left, that when you're 90 years old, that your 27-year-old beau is going to impregnate you. And that's because this process happens and ends after, you know, before birth, or sometimes a couple of months after birth. You know, you have some eggs. So anywho, this primary oocyte is diploid, so that means it has 46 chromosomes, and we need to reduce those chromosome numbers by half, because egg and sperm need to only have 23 chromosomes so that when they come together, boom, they make 46. So in meiosis, we still have meiosis 1 and meiosis 2. Between the 3rd and 7th months of fetal development, those daughter cells or primary oocytes prepare to undergo meiosis. They proceed as far as prophase 1, but then it's going to stop and come to a halt. So we have our primary spermatocyte, and as it goes through its division, the primary oocyte remains in a state of suspended development, so it's not going to go anywhere else until it reaches puberty. So it's going to kind of just hang out here and stay here until it reaches puberty, when the levels of the hormone follicle-stimulating hormone have triggered the start of your ovarian cycle. So right now, for my girls that have not had their first menstrual period, they're stuck in primary oocyte stage, and it's kind of just suspended there in meiosis 1. So it hasn't completed that process of meiosis, it's just kind of stuck there. And remember, it's just, you know, this is the beginning of it. We started meiosis during fetal development, prior to pregnancy, prior to being born. Each month after the ovarian cycle begins, some of the primary oocytes are stimulated to go further development. So meiosis 1 is then completed. So at the end of meiosis 1, so this only happens for one egg at a time. So this particular egg, or few, very few, eggs at a time, is going to be stimulated to undergo further development. And when it separates, remember meiosis has two divisions, meiosis 1 and meiosis 2. First time, one cell divides, becomes 2. Second time, those 2 cells divide, and you have a total of 4. So we're at that first stage there, where this one cell is going to become the 2. Now, when it becomes 2, one of them is going to be a polar body that we call our first polar body, and then our other one is going to be the secondary oocyte, which is still diploid, still has 46 chromosomes that will continue on through meiosis II. The secondary the polar body doesn't complete meiosis II. It usually doesn't complete meiosis II. So this is really just taking up excess chromosomes. There's not really going to be much that happens to it. It's just not functionally going to be an egg. So each month after ovulation begins, one or more secondary oocytes leaves a suspended phase, a metaphase, meiosis II. So we have another pause here. So the secondary oocyte is released, and it is a mature ovum, and it's going to be haploid now. So after we've separated and pulled apart, so that after, we have that metaphase that's happened there, it's going to stay suspended and wait. So after we've had that process take place, it's going to wait until there's fertilization that takes place. So these over here are also our polar bodies, and then as we had this secondary oocyte go through meiosis II, we release a secondary oocyte released in metaphase of meiosis II. We have a second polar body, and we have the ovum here that is now a more mature. So the polar bodies that are produced in meiosis I may or may not undergo meiosis II, but at any rate, any of these polar bodies, and their whole job is just to take up excess chromosomes, is that they're going to eventually disintegrate. And then we'll stay here until fertilization takes place. So meiosis II will be completed only if fertilization occurs. If fertilization does not occur, then this ovum will disintegrate into the wall of the uterus, and it will be sloughed off during the menstrual cycle. All right, so that's a nice segue into the female reproductive system. Female reproductive system produces sex hormones such as estrogen, estradiol, progesterone, and also functional gametes. Those would be those oocytes that we just looked at. Female reproductive system also protects and supports the developing embryo and nourishes the inborn infant through the mammary glands as a route of breastfeeding. Now, obviously, you don't have to breastfeed to nourish your newborn infant, but there are lots of benefits to breastfeeding, and a lot of moms choose to breastfeed, and your body is actually made to do it. Structure of the female reproductive system: ovaries, fallopian tubes, aka uterine tubes, the uterus, the vagina, and then you have your external genitalia, your labia, minora, and majora are part of that external genitalia. Oogenesis, which we just looked at, is the process of producing an oocyte or an ovum. Remember, this process actually started before birth, had a little bit finishing up kind of right after you're born, first couple of months of light. It accelerates at puberty, so remember that when we go through that ovulation cycle, that process is going to result from follicle-stimulating hormone being released from your pituitary gland. It's going to, trigger the release. It's going to affect your ovaries, and that hormone will allow for those eggs that were arrested in meiosis I to complete that process of meiosis I and then go through most of meiosis II. And remember, it doesn't finish completion unless that egg is fertilized. And then this process of oogenesis ends at menopause. Basically, you ran out of eggs, so you're all done with your eggs. So you're all done with your menstrual cycle. You're all done with ovulation. In fetal development, this process of oogenesis, remember we said between the 3rd and the 7th month, the primary oocytes prepare for meiosis, and they stop at about prophase I of metaphase. Atresia is the degeneration of the primordial follicles, so that when you're born, you have about 2 million primordial follicles at birth, and each one of them has a primary oocyte that when you go through puberty, once that follicle-stimulating hormone is released, it's going to trigger some of those 2 million primordial follicles to complete meiosis I and go through meiosis II. By the time you get to puberty, that number drops drastically, and most folks only have between 400 and 450,000 eggs. Some people have a few more, some have a few less. And of those 400,000 eggs, a couple of them might get fertilized. And once again, it's kind of repeating the same information. Primary oocytes remain in suspended development until puberty. At puberty, because of those rising levels of follicle-stimulating hormones, since we have said the name of this hormone so many times, I want you to remember what it does in the male and in the female. For both of them, it helps to trigger the process of oogenesis or spermatogenesis, which is gamete production. Each month thereafter, some of those primary oocytes are going to be stimulated to develop further, and remember, they will go all the way through to metaphase of meiosis 2, where you will have these haploid cells. Big take-home message here from what we've talked about on a cellular level between males and females in this process of mitosis and meiosis. Males, for every mitotic and meiotic event, will have 4 functional sperm. Females, for every mitotic and meiotic event, will only have 1 functional egg and several polar bodies. That 1 functional egg is still going to be haploid and have 23 chromosomes. And in addition, males can continue this process of meiosis and mitosis to make functional sperm throughout their lifetime, whereas women, the process started before birth. So we have our set number of eggs that we're going to have prior to being born. Maybe we'll make, a little bit of it finishes up after you're born, but for the most part, the number of eggs you're going to have, that's determined before you are born, and that you don't complete, for the male, they complete mitosis and meiosis completely for those 4 functional sperm, but for women, this very precious, energetically expensive ovum, it takes, we have 2 parts where the development is arrested. The development is arrested prior to puberty, and it's arrested and at the beginning or the end of mitosis, beginning of meiosis, and then it's arrested again in the middle of meiosis 2. So we have 2 points of pause for oogenesis. The ovarian cycle does not take place until after sexual maturation. Remember, follicle-stimulating hormone has a lot to do with that. A different group of primordial follicles is activated each month, and they're divided into 2 different phases. The ovarian phase is divided into 2 different phases. We have our follicular phase, which is the pre-ovulatory phase, and the luteal phase, which is the post-ovulatory phase. The ovarian follicles are structures within the ovaries where the oocytes grow, and meiosis 1 takes place within those follicles. So each one of those primary oocytes is going to be found within a follicle. They are located within the ovary or the cortex of the ovary, in the middle of it, not on the kind of on the outside of it, rather not in the middle of it. These clusters where these primary oocytes are called, we call them egg nests. For fertilization to take place, if that primary oocyte goes and finishes meiosis 1 and gets all the way to meiosis 2 and kind of stops in the middle of meiosis 2, that egg is going to be released. When that egg is released from the ovary, it travels through the fallopian tubes or the uterine tubes, and it just kind of slowly makes its way down. Now, in order for that egg to be fertilized, the secondary oocyte has to meet a sperm during that first 12 to 24 hours as it makes its way down from the fallopian tube to hang out in the uterus. Fertilization usually happens at the first 1/3 of the fallopian tube, which is between the ampulla and the isthmus of the fallopian tube, and these are 2 structures that you should be familiar with, as where you would find that. Right there in that first portion is where you'd find fertilization, where it takes place. In the fallopian tube, just like the sperm had a seminal vesicles, prostate gland, and vulvourethra gland that made these secretions that helped to make their journey more tolerable. The fallopian tubes also provide a nutrient-rich environment and secretions from these PEG cells in there that contain fats and sugar to help nourish this egg as it makes its journey or lays in wait for a sperm to come and fertilize it. And if that fertilization takes place, you obviously want to make sure that this fertilized egg has enough nutrients and lipids to sustain it as it makes its way to the uterus and implants itself into the wall of the uterus. Those nutrients supply the spermatozoa and the developing pre-embryo the needed resources to continue this journey and to do its thing, to live and to be viable. Uterus is where your developing embryo is going to hang out. Provides the developing embryo weeks 1 through 8. We call it an embryo, and then from weeks 9 through delivery, we call it a fetus. The fertilized egg attaches itself to the wall of that uterus, provides mechanical protection, nutritional support, and then it also removes waste. So if that egg is not fertilized, it will disintegrate into the wall of the uterus, and part of your menstrual cycle is the sloughing off of that functional layer of the uterus, of the endometrium. So we talked about in lab how you have that functional layer and you have that basilar layer that's right behind it. Functional layer is the part that you lose off via during menstruation, if you're fortunate enough to have a uterus. So the endometrium consists about 10% of the uterine mass. Glandular and vascular tissue support the physiological demands of a growing uterus, with lots and lots of blood vessels that are there, lots of glands that will be the delivery method through which hormones that are required to develop that fetus, or that growing fetus, if there is fertilization that takes place. These uterine glands open onto the endometrial surface, and once again, it's just to provide the developing fetus the hormones and the necessary factors and proteins that it will need to continue to develop. Estrogen is the hormone that causes the uterine glands, blood vessels, and epithelium to change during the phases of your monthly cycle. So whether fertilization takes place or not, you have these uterine glands that are there to kind of monitor and dictate the activities that happen in the uterus, whether fertilization did take place and we are doing all this to support the developing embryo, or fertilization did not take place and we need to maintain the health of the uterus by getting rid of the waste, i.e. the additional layers of the uterine wall that have been fluffed up through this uterine cycle, through this ovulatory menstrual cycle, and then the egg that's disintegrated in there. We need to kind of clear all that space out so that we can prepare the body again for next month, just in case fertilization takes place. Uterine cycle is also called your menstrual cycle. Lasts 21 to 35 days. The average is 28 days, and some people, such as myself, are super irregular. So sometimes it's 21 days, sometimes it's 27 days, and sometimes it can be 35 days. It just does its own thing. Responds to hormones of the ovarian cycle. Menses, or the proliferation phase. When that's happening, the ovaries are going through their follicular phase. So there are two things going on here. We have the ovaries have their thing that they're doing, and the uterus has its thing that it's doing as well. And when you're having that menses, or the proliferation phase of your uterine cycle, then the ovaries are in the follicular phase. So that's that pre-ovulatory phase. And your secretory phase that occurs during ovulation, luteal phase, or the post-ovulatory phase. So ovulation is, dropping an egg, moving that egg along. So there are different things that take place, and I'm going to highlight that I want you to remember this sort of thing. That there are two phases, two organs in the female reproductive system that have their own phase, thing that's going on, and what's happening in each of those phases. Menses is the degenerating tissues break away and they enter the lumen of the uterus. That functional zone of the uterus is lost and it exits out of the external os and through the vagina. And remember, only the functional zone is affected. That basilar zone is supplied by arteries, and you're not going to lose that basilar zone of your endometrium. So remember, if we're in menses, then we're in that pre-ovulatory phase in your ovaries. So remember those two phases we had. We had the pre- and post-ovulatory phase, or we had the follicular phase and the luteal phase. Follicular phase is happening in your ovaries while your uterus is going through menses, or the actual sloughing off of the functional zone of the endometrium. The process of menstruation is endometrial sloughing, we talked about. It lasts about 1 to 7 days. Remember that changes for some people month to month. It changes for some people period. Not everyone has the same length of a period. Some folks are always 3 days, and they're always the same time of month. Their cycle is always 28 days. Some folks, sometimes it's 7 days, sometimes it's 5, sometimes it's 3, sometimes it's always 7 days. So it just depends. Proliferation phase, the epithelial cells of the uterus gland will multiply and spread across that endometrial surface, and it restores the integrity of the epithelium. We have more vascularization and more growth that will restore that functional zone in this proliferation phase. So we are increasing the wall of the endometrium, and we're replacing that functional zone that we lost off during menstruation. Now note here, at the same time that this is happening, there's enlargement of the primary and secondary follicles in the ovary. So we are going through this process of ovulation. So in menses, we're in the pre-ovulatory stage. In the menstruation, we're still kind of in pre-ovulatory phase, but we're kind of beefing up for ovulation. But now, when we're proliferating in the uterus, now that we've gotten rid of that old endometrial lining of the functional zone, we're rebuilding more endometrial lining for that functional zone. And we're in the ovaries, so the uterus is preparing, you know what, we might get fertilized this month, we might have fertilization, we want to make sure it's nice and soft and cushy, and we have enough blood supply and enough nutrients to support this fertilized egg if it does happen. So that the ovaries, they're beefing up to release this egg. So we're enlarging the primary and secondary follicles in preparation for this egg to be released. In the secretory phase, the endometrial glands enlarge, increase their rates of secretion. The arteries in the uterine wall are going to get longer, and they go through the spiral zone. This begins at ovulation and persist as long as the corpus luteum remains intact. So in your ovary, remember those primary and secondary follicles were getting larger? At this point here, they have released the egg. So the egg has been released, and what's left over, that follicle is not called a follicle anymore, it's kind of scar tissue, and we call it the corpus luteum. So that corpus luteum is going to release a hormone as well called progesterone. And this hormone, progesterone, helps to make sure that the uterus stays nice and fluffy, just in case that egg that we released 12 to 24 hours from now, it might come into contact with the sperm. And if it does, we want to make sure that we're prepared to receive it. So the secretory phase peaks about 12 days after ovulation, and then any time after that, the glandular activity is going to start to decline. So this process of increasing the rate of secretions and in the uterus and the blood vessels going all through that functional zone, that lasts about 14 days. At that time, in the ovaries, the corpus luteum is going to stop making the stimulatory hormones, which in part help for this phase of the uterus to do its thing. And the hormone in particular is progesterone. The Menarche is your first uterine cycle, happens about 11 to 12 years old. Some people are late bloomers, like my mom. She didn't have her first Menarche until, I think she was 20 years old, 20 or 21 years old. I didn't start till I was 16, so some of us are late bloomers. Menopause is the termination of that uterine cycle, and that happens between 45 and 55 years of age, and some people it's a little bit later than that. My grandmother didn't go through menopause until she was 70, I believe, and my mother, who is in her 60s, still hasn't gone through menopause yet. So we start late, we end late, I guess. Amenorrhea is a failure to, so there's two types of it. You have primary amenorrhea, or you have transient secondary amenorrhea. That's just a failure to initiate menses. That could be as a result of chromosomal abnormalities. You only have one X chromosome or something like that. Transient secondary amenorrhea usually is your period stops. We can see that for people, girls that run track or cross country or do marathons, things like that, their menstrual cycle will usually stop in season. I know when I ran track, while I was track season, I didn't get a period. Physical or emotional stress can cause this. There are also hormonal changes that can happen with your pituitary gland. If you have tumors on your pituitary gland, it can cause transient secondary amenorrhea. And if that's the case, the tumor can be removed, or they can give you drugs to shrink the tumor. In that case, or even emotional stresses can cause you to not have a menstrual cycle. All right, and the last thing we're going to talk about is your vagina, if you have one. Function of the vagina, get rid of menstrual fluids, so that functional zone that we talked about getting rid of. It also receives the sperm during sexual intercourse, and it also forms the bottom portion of the birth canal. All right, so I guess we'll finish this one up, and that way we could be all done talking about a vagina. There we go. So in your vagina, you have bacteria that are part of your normal flora, your normal microbiota. They're just your residents there, and they're meant to keep your vagina acidic. So it creates an acidic environment of the bacteria. As a result of it being more acidic, it's going to resist the growth of many other bacteria. And the way that you support your bacterial that live in your vagina is through the different nutrients that come from your cervix. So the bacteria that are in there, they should be in there. A lot of the bacteria that are found in yogurt are the bacteria that are in your vagina. So if you're taking broad-spectrum antibiotic, or if you have reoccurring yeast infections or prone to yeast infections, because yeast kind of hangs out in there too, but not nearly as much yeast as bacteria, and the bacteria help to keep those yeast numbers in check. However, if you have reoccurring yeast infections, or you're taking a broad-spectrum antibiotic, it's always a good idea to have a diet with yogurt, because that helps to replace those bacteria in your vagina that should be in there, and they will outcompete the yeast and keep those reoccurring yeast infections from taking place, and definitely help act as a preventative measure from developing yeast infections if you're taking a broad-spectrum antibiotic, which has a tendency to get rid of all your bacteria, even the good guys. A vaginal smear is done in a pap smear, or it can be done as a pap smear, is the sample of the vaginal cells to look to see if there are any damages to those cells, and it can also tell you about where you are in your ovarian or your uterine cycles, or both of them. All right, so now we're finished. So in lab tomorrow, we're going to do a rat dissection, and that'll be cool. We'll look at some rat parts. So we'll do a rat dissection in lab, and then next Thursday, remember, you'll have your lab practical. On Monday, pretty good chance that we will finish this discussion up, and I will also be posting for you sometime this weekend or sometime later this week another video that I like for you to watch, and it's about life, the miracle of life, I think is the name of the video. So we'll be looking at that too as we finish talking about the female reproductive system. All right, and once again, I am so sorry that I was like 25 minutes late getting in here, almost a half an hour late getting in here. Collaborate just would not work for me the way I wanted it to. But at any rate, we got it all done, and I will see you guys actually in class tomorrow. Have a great hump day. Bye.